Now we are coming to PCR test for RNA. So far we have studied PCR test for DNA. Now if it is a RNA, because when it is going for viruses, viruses are having any one nucleic acid. All other organisms are having DNA as well as RNA, but viruses either RNA or DNA will be there. Assume that one virus is there. That virus is having only DNA. PCR test, previously DNA test we have taken. Suppose one virus we are taking, it is harmful, it is causing disease to our body, but that virus is having only RNA, no DNA. Then we have to go for PCR test for RNA. Now for PCR test for RNA, reverse transcriptase PCR test is done. Now reverse transcriptase is a type of enzyme. Now reverse transcriptase PCR test is done. So that short form RT means reverse transcriptase PCR test. That is polymerase chain reaction test. Okay, this is done. Now in this messenger RNA we are taking RNA. Three types of RNA will be there. One is messenger RNA, second one is transfer RNA, third one is ribosomal RNA, of which messenger RNA is very important. That is only carrying the message. Now this messenger RNA is a small fragment of RNA you are taking, mRNA. It is changed into complementary DNA. Now that small fragment of RNA is changing into DNA. Now see that complementary DNA. So RNA is changing into DNA. Okay, that is done by reverse transcriptase enzyme. By the enzyme reverse transcriptase. Okay, so in this test, messenger RNA is converted into complementary DNA. Right. PCR in, that's all, only this. Now after doing this complementary DNA, the complementary DNA copies can be identified, multiplied. Then we can make the test. Now by this PCR test for RNA is over. Now we are taking PCR in clinical diagnosis. Diagnosis already I told you, okay, checking whether the individual is having any suffering. That is called a diagnosis done in the medical labs. Clinical. Suppose the person is having infection, okay. Now doctors are checking. After that they are suspecting the patient. Now that is called a clinical diagnosis. They are suspecting it may be bacterial infection or viral infection. That is called. At the time, how the PCR test is done. So this PCR test in clinical diagnosis is used to diagnose genetic disorders. Whether the person is having any genetical hereditary problem or genetic disorders are there. Viral diseases, whether the infection is there, viral infection, bacterial diseases, etc. All these things can be identified. The PCR is used to identify a particular pathogen. What type of pathogen is present? First infection. After that, what exactly this pathogen, whether it is influenza virus or it is HIV or it is polio virus, anything they can identify, or even the what is that virus which is causing this, now coronavirus, even that coronavirus is a particular pathogen they can identify. Then the traditional method involves, how the traditional means old method, how it is doing, it involves culturing the infected microbes. Now already the infection is there. Now they will collect the microorganism. They will culture the microorganism. That means making the microorganisms to multiply, culturing the micro, and performing metabolic. Metabolic means chemical reactions. And other test, they will do chemical reactions test and other test to identify the microbe. But it is little tedious. Accuracy may not be there. But PCR will give accurate information about the microorganism. PCR test is performed to find out the presence of DNA of the microbe, not the microbe, DNA. Even the DNA will be identified from the clinical specimens. What is the clinical specimens? Now the blood is a clinical specimen, because the doctor is telling blood sample, you give that. We are giving little blood. So the blood is a clinical sample. Stool, that is undigested food. Okay. Then sputum, when we are having a respiratory infection, whenever we cough, the mucus is coming out. It is called a sputum. Spinal fluid in the spinal cord. and also around the brain, a fluid is present, colorless fluid. It is called a spinal fluid, the spinal fluid, etc. They are all clinical specimens. Specimen means, don't think that some organisms they are. The specimen means the sample what we are giving, what is taken from our body. Okay, from that also PCR test can be done. Now, PCR in clinical diagnosis, previously for the test, now diagnosis. It is also used in prenatal diagnosis. What is the meaning of prenatal? Diagnosis means identification. Okay, now finding out. Prenatal means before the birth. Okay, natal means birth of the child. Pre means before. Prenatal. It is also used in the prenatal diagnosis of hereditary diseases. For example, when the child is growing, we should not use the term child, we have to use fetus. When the fetus is growing in the mother's uterus, okay, the growth is going on, that is fetus I told. But at the time itself, when the child is growing, when the fetus is growing, is there any hereditary diseases we can identify with the PCR? Okay, we can identify. So, in the fetus, from the chorionic villi. Now, that means the fetus is covered by 3 membranes. One is called the chorionic villi, one membrane. Or the cells from the amniocentesis. The amniocentesis is the fluid. Okay, that is a amniotic fluid. It is having a lot of cells. Either taking the cells from the amniotic fluid or from the chorionic villi. We have to identify the chromosome, whether any defects are there. If the defects are there, hereditary diseases are there, that we can identify. Next one is hereditary diseases like sickle cell anemia. What is that sickle cell anemia? Normally, we are all normal people. When we take our blood, where the RBCs are there, red blood corpuscles, RBCs. The RBCs are circular, they are plate-like and biconcave. That is a normal RBCs. But for some people, the RBC will not be circular. Okay, the RBC will not be circular. The RBC will be irregular and microscopic sickle shape, sickle. Now, the sickle is an instrument used by the farmers to cut the crops. Okay, it will be a metal plate which is bent like this, lot of teeth-like structures will be there. Like that, the RBC will be present. Such type of RBCs we can call as a sickle cells. Okay, that condition is called, the disease is called the sickle cell anemia. It is a hereditary disease. Phenylketonuria. It is a problem in the urine formation. Now, in our urine formation, one chemical reaction is there, urea formation. In the urea formation, what is that? Metabolism. It is incomplete metabolism. At the time, instead of urea, phenylketonuria, another one compound will be formed. It is poisonous chemical. Okay, actually urea is a less toxic, but this phenylketonuria is very, very poisonous, which is damaging the kidney, damaging the urinary bladder, everything. So, that problem, it is also hereditary. Beta thalassemia. Now, beta is the type. Thalassemia. Thalassemia means it is a problem in the what is that? Immune system. That means bones, marrow problem will be present. As a result of which, the immunity development is not at all right. That is called beta thalassemia, etc. Can be detected. Okay, the next one, viral diseases like cervical cancer, cancer in the cervix region. In the female reproductive tract, we can see this cervical region. So, the cancer will be developed, can be diagnosed by this method. Then cDNA. Okay, what is this C? Complementary DNA. Just before in the previous slide we have studied, the messenger RNA is changed into complementary DNA. Now, that only complementary DNA is a valuable tool for the diagnosis of retroviral diseases. The diseases caused by retrovirus. Retrovirus is a group. It is not single virus, it is a group. Now, some diseases and bacterial diseases. I will give one example for retroviral disease, HIV, retroviral. Okay, cancer causing virus. Viruses are causing cancer. Now, that is called a retrovirus. These are all the retroviral diseases and the bacterial diseases. Next, sex of a human, livestock, etc. can be determined by in vitro fertilization by fixing specific genes in the sex chromosomes. Okay, so the sex of the human can be determined. Now, actually, natural phenomenon, if you take it as natural, naturally the sex of the individual will be either male or female. But artificially, by using this as PCR, we can make the individual to become either male or female, not natural, artificially. The sex of the individual can be determined artificially. Livestock, what is the meaning of livestock? Okay, the domestic and economically important animals are called livestock. Say some animals are domestic, pet animals, but they are not economically important, but they are pet, that's all. But economically important animals like sheep, goat, okay, that is a hen, that is poultry, then cattle, everything. Now they are economically important. So everything together we can call as livestock. So human, sex of human and a sex of livestock, etc. can be determined by in vitro fertilization. Okay, what is that? In vitro fertilization means the fertilization process is taking place in the laboratory by fixing specific gene in the sex chromosome. Even the sex chromosome we can alter, and the genes present in the sex chromosome can be altered. Okay, sex linked disorders. Now there are certain hereditary diseases. These hereditary diseases are coming from parents to the offsprings. Generation after generation the diseases will go, and they are related with the sex of the individual. Only males will be affected by some diseases. Only females will be affected by some hereditary diseases. They are called sex linked disorders, diseases. Could be diagnosed by zygote. By this diagnosis, from the zygote itself, zygote means fertilized egg. From the fertilized egg level itself, we can identify, is there any sex linked disorders? Suppose the sex linked disorders are there, or any other type of hereditary disorders are there. Already we have studied gene therapy. Okay, now the defective genes will be there. Now we have to remove the defective genes and we have to keep the correct gene, then the disorder will be disappeared in the subsequent generations. Now applications of PCR. Okay, PCR is a test. Where at all we are applying? In which fields we are applying? That we have to see. To study the differences between genome of two different organisms. What is genome? The amount of DNA present in one organism cell, one cell. Okay, for example, in our body, millions and millions, billions and billions of cells will be present. But we have to take only one cell from our body. Now, either one nucleus is there. In the nucleus, 46 chromosomes are there. In the 46 chromosome, how much amount of DNA is there? That is genome of human. Okay, for example, we have genome of human being. And also, we are having another one entirely different organism, monkey. Now, it is having 44 chromosomes. Okay, in every cell. Now we have to take the 44 chromosomes and we have to see how much amount of DNA is present. Then we are comparing the DNA of human, DNA of monkey. That is called a comparison. Okay, the study to study the differences between the genome of two different organisms. Okay, there is a done. Now it is used in phylogeny. Phylogeny is the study of evolution. How man came from monkeys. Now we are telling that scientifically we are proving that man population, human population originated from monkeys population. That means some relationship is there between the DNA of monkeys and DNA of human being. What are the relationship? What are the differences? That also we can. That is phylogeny. What is that? Evolutionary sequences. With the help this is applied. That is PCR can amplify the minute quantities of DNA from hair, mummified tissues, bones, blood, semen, fossil materials, etc. For example, a fossils, I'll tell you. One particular population of or one particular species of animals lived in the past. Okay, but they are not living now. The whole population is vanished. Okay, long time before, maybe 1000, 2000, 3000 or 1 million years before they lived, but now they are not living. But when we are digging the soil, we are getting fossils. The dead remains, maybe bones or hairs or nails, claws, something will be there that is left in the soil. The flesh and all decomposed, but only hard materials will be present. They are called fossil materials, but they are also made up of cells. Assume that we are getting one small piece of fossil, then in that cells will be there, but in the cells we can find the DNA, little quantity of DNA we can take. But what we can do is that little amount of DNA we have to take, then we have to make this PCR test. We have to multiply the DNA, enormous copies we can multiply, and we can find out what are the characteristic features of the animals lived in the past. That is, so the DNA from the hairs of the dead animals, mummified tissues, mummies. Okay, now that they are processing the dead body of a human of once upon a time, long time before, and they used to keep it in the mummies, okay, preserved. Lot of preservative they used to. But now the bodies will be there. So from the body, the cells or tissues we have to take from the DNA can be extracted. DNA can be extracted from bones, blood, semen, that is male germinal cells, semen, fossil materials, etc. Okay, it is used in the field of forensic medicine. Okay, fingerprints, forensic medicine, even in the forensic. Suppose the fingerprints are like this, means what will be the DNA? We can easily trace out. Now for that, the PCR is very useful. It is used to develop the DNA fingerprinting. Okay, for criminals, particularly for criminology. The criminals, what they will do, they will take the fingerprints and they will identify the DNA. Okay, and whether the DNA is matching with the person or not, they can easily identify it. Used to develop DNA fingerprints in criminology. It is used in gene therapy. Just before I told you. Now in the individual, in the zygote itself, we are finding in the in the cells, okay, in the chromosomes, some defects are there. Then the defective genes must be removed and the corrective gene should be kept. So it is used in the field of gene therapy. Okay, now we are coming to the next new topic. Okay, it is another one, technology we can say, transgenic animals. Okay, transgenic animals. Now it is a new area. It is also fast improving, and a lot of new varieties of animals are developed with the help of this technology. Transgenic animals. Okay, what is a transgenesis? We have to see. This technique is used to bring new varieties of animals. See that by applying this technique, we are getting new varieties of animals or hybrids. Hybrid means the new variety. But the new variety is developed from entirely two different species of organism. That is called the hybrids of economically important animals. The animals which are giving us some product. They are called economically important animals. So in the economically important animals, hybrids can be developed or new varieties can be developed. Then in the traditional method, long time before the development of science and technology, the people, the farmers, the people and all, they used to bring the new varieties, but simple method. It is called selective breeding method. Okay, the method is called selective breeding method. I'll tell what is the selective breeding method. They want genetically stronger individuals. But what they will do is, in the area, one cattle, for example, one female cattle, it is having good qualities, lot of good qualities are present. Maybe in another one area, the male animal, male cattle, bull, which is having extremely good quality, there is characters, okay, good qualities, body characters we have to see. Then we have to allow the two better varieties or the best varieties to mate each other. That is called a selective breeding. Breeding means reproduction. We are selecting the individual, male and female, we are making them to mate each other to bring this, crossing the animals with a good genetic characters, to improve the genetic characters in the subsequent generation. That was the old method. So generation after generation, they will produce strong varieties of organisms. Now we will take transgenesis. What is the meaning of transgenesis? You have to see. It is a technique. Transgenesis is a method of introducing, is a technique of introducing foreign gene DNA fragment. What is that foreign gene? The gene taken from entirely different species. Foreign gene DNA fragment into the genome. Genome means the whole amount of DNA of the animal to create, to develop, to bring out stable, heritable new characters. Stable. And it should not be temporary. It should be subsequent generation. Stable, heritable. That good quality character should go on to the subsequent generation, new characters. Okay. Transgene, we have to know what is the meaning of transgene. Is the foreign gene which is taken from the other organism, entirely different species. We are telling, no, transgene is the foreign gene that is introduced into the recipient animal, which is given to one particular animal. Then transgenic animal is the one which received the foreign gene. Then we are telling what is a transgenic animal. It is a cattle. or it may be any other economically important animal, but it has received the foreign gene. Foreign gene means not coming from some foreign countries. The gene has come from entirely different species of another one animal. That is called a foreign gene. Transgenic animal is the one which received the foreign gene. It is also called genetically engineered or genetically modified animal. So transgenic animal is also called genetically engineered, because the scientists, biotechnologists, they are doing this work. And genetically modified, it is changed, genetically modified animals. Now we are coming to steps involved in the production of transgenic animals. Okay, to bring one transgenic animal, what are the steps we have to do? What are the steps to be taken in the technology? We have to say identification and separation of desired gene. What is the desired gene? The good quality gene. First we have to identify which animal is having, in which chromosome that good gene is present. Desired gene means good gene, where it is present. We have to identify and we have to separate only that particular gene. I'll give one example. One cow. Okay, that cow is giving tremendous amount of milk. So the milk production is extraordinary. So the gene for milk production is good quality gene. Now we have to take the gene. Okay, that is the desired gene. So the identification of such gene, separation of the desired gene. After that, selection of the vector. But whenever you take a gene from one organism, when we are trying to introduce into another, the gene will not directly go. We need a vector. Vector means a carrier. A carrier should be there. The carrier only can take that to the other cell. So selection of the vector. Which vector is suitable? Generally the virus DNA. Generally virus DNA is taken as a carrier. Okay, generally a virus DNA or direct transmission. Sometimes directly the DNA will enter. Okay, direct transmission can be done. Either direct transmission, if it is not possible direct transmission, through vector, we have to send the desired gene to go to the other organism. Combining the desired gene, I told you, the good gene, desired gene with the vector. First we have to make the desired gene and vector, they have to combine together. So the good gene should be connected with the vector. Vector means carrier. Then introduction of transferred vector into the cell. Then once the vector is connected with the desired gene, it should be transferred into the cell of the other animal. Okay, cells. Or it can be entered into tissues or into the embryo or matured individual, anywhere. Into the body cells, into the tissues or into the embryo or into the matured animal, the vector with the desired gene should be introduced. Then demonstration of integration and expression of foreign gene. Okay, that foreign gene, the one which is taken from one organism, that desired gene is a foreign gene. And we have to prove that whether it is integrated with the new organism and whether the character is expressed. We have to demonstrate it of the foreign gene in the transgenic tissues of animals. Now transgenic animals such as, which are all the transgenic animals? Already they did lot of transgenesis in many type of animals. One is mice, next is a rat, okay, rabbit, pig, cow, goat, sheep, and also in fish have been produced. So transgenic animals, good quality animals are produced. So the animals which are bearing the foreign genes in their body and producing good quality characters, we can call as transgenic animals. Now we are coming to biological products and their uses. Okay, by biotechnology, what are the biological products we are getting? Now whenever we are doing some research, the research is giving us some benefits. Okay, excellent benefits. Now they are called products. And what are the products and what are their uses? How far these products are beneficial to us? That we have to. That is, all the products must be useful for mankind. Okay. These products are obtained through biotechnology from microbes. From the microbes we are extracting animals and plant cells. If there is much demand, the products demand should be there. Then only we can produce this. Suppose there is no demand, there is no need. Producing these products by biotechnology, applying biotechnology is waste. So which are all the products are in demand? Those products we can obtain from this. It is used for the prevention and treatment of diseases. Biotechnology is useful to prevent the disease. For example, vaccine. Already in biotechnology we have studied about recombinant vaccines are prepared. Why the vaccines are produced? To prevent the disease. The disease will not be allowed to expressed in the individual's body. Prevention. Okay, it is already developed. Disease is expressed. Then treatment of the diseases. Then the products include, what are the products we are getting from the various microorganisms, animals and all? That is by biotechnology. First one is antitoxin. Now, antitoxin means the chemical which is spoiling the poison produced by the microorganism. Antitoxins. Bacterial and viral vaccines. Okay, they are all the product. Just now I told vaccines are produced. Bacterial vaccines, viral vaccines. Monoclonal antibodies. Antibody normally produced by our blood. There is natural immunity, but artificial immunity. We have to take the microorganism, we have to introduce into the other microorganism, the genes must be introduced and we have to multiply that. From there we are extracting antibodies, from the microorganism we are extracting antibodies, which is called the monoclonal antibodies. Proteins, for example, sometimes the child is suffering with the protein deficiency. Now we can extract protein, same type of protein from microorganisms and other plants. Proteins, blood products, okay, blood products. Now synthetic hemoglobin, okay, synthetic bone marrow, everything can be there. That is blood products. And hormones or extract, hormone extracts. All the things can be the products. So what are the products we can say? Antitoxins, bacterial and viral vaccines, monoclonal antibodies, proteins, blood products and hormone extracts. Okay, hormones and antibodies. Now antibodies and the hormones are produced commercially. They are produced commercially in enormous quantity in medical industries. Now medical industry means pharmaceutical industries. For example, what is that? Hormones, insulin hormone, very demand, high demand is there because lot of people are now diabetic. Okay, now more than 15 to 20% of population, human population are diabetic. They need a hormone injection. What is that? Insulin injection. There is a hormone we are extracting and that is used, they are manufacturing in medical industries. Antibodies, monoclonal antibodies, okay, because nowadays the human what we can say immunity level is going down, so we need support from outside, that is monoclonal antibodies. Next, recombinant human insulin, this is what we studied in the first part. Growth hormone, vaccines, human alpha lactalbumin. Lactalbumin is the protein, a type of protein, are available today. Now all these things are available with the help of biotechnology. Animals are used as bioreactors. Now animals are used to produce these products. Animals are used. So such experimental animals are commonly called bioreactors. Now experimental specimen, experimental animals to produce desired proteins, monoclonal antibodies, etc. Now monoclonal antibodies are used to treat. Why these antibodies are there? To treat cancer, okay, heart diseases, any heart problem, and a transplant rejection. What is a transplant rejection? We have to see. Sometimes one person is losing both the kidneys. Okay, both the kidneys are collapsed, damaged. So doctors will say kidney transplantation must be done. Okay, both the kidneys are removed. Someone will donate one kidney. Out of two kidneys, one kidney is donated. He is called a kidney donor. Okay, that donated kidney is given to this person from whom two kidneys are spoiled, they'll do. Now this is called a kidney transplantation surgery, they will do. Okay, once the kidney is transplanted, many people, the transplanted kidney will be accepted by the body of the recipient. Okay, accepted means no problem. For some people, the transplanted organ is not accepted, it is rejected. It is called a transplant rejection. So what will happen? The body which received the kidney will try to spoil the kidney which is received. So that is called a transplant rejection. For that, monoclonal antibodies are very essential to preserve, to protect the transplanted organs. Natural protein adhesive. Now this is very important. Adhesive means any substance which is gum-like, which is useful to join. Paste. It is a paste. Now it is a protein paste. Natural protein paste. Okay. Now we have to see natural protein adhesives are used to reattach tendons. Now, in the field of medicine, a new term is given. Now, the new term is nothing but fevicol, human fevicol. But actually, the fevicol is a common term. Now, whenever the woodwork is done by carpenters to join the wooden plates, they are applying fevicol. They are applying fevicol. Okay, they join. Wooden plates will be joined very well. They are attaching very, very strongly. But this natural protein adhesives are called a human fevicol. Now, whenever a tissue or organ is damaged or cut, the edges will be sutured previously. Sutured means they will stitch it. But now they can apply this natural protein adhesive so that the part will be there. I'll give one simple example for natural protein adhesive. Imagine that by accident, with the help of a glass piece or with the help of knife, the skin is torn little deeper. Okay, lot of blood is coming. Now, previously, old method, even now also, they will make the skin edges to join and they will make the sutures, maybe 5, 6, 7, 8, 9 sutures they will do. But now it is very simple. They will apply this adhesive, protein adhesive, the gap where the cut is there. Then they will bring the two edges and they will hold it for some time. Then automatically they will attach. Okay, that is natural protein adhesives are used to reattach the tendon. Tendon is nothing but the ends of the that is what muscles. A strong cartilage will be there. That is called a tendon. Okay, even at the end of each bone we can find the tendon and the tissues. To fill the cavities in the teeth, sometimes the infection will be there, cavities will be formed. There also this protein adhesive can be applied. And to repair the bone fractures. Now once again, fracture, bone is broken. But normally what they will do, they will keep that bones in correct position and they will put bandage. They will give the support and they will put the bandage. It is normal method. But what they are doing now, once the bone is broken, the broken edges, where the broken edges are there, they are applying this protein adhesive. Okay, they are applying and they are bringing the two bones in proper position. They will support and put the that is what bandage. So within a few days, not necessary to wait for about 15 days, 20 days, 30 days like that. Within a few days, the broken bones will be strongly attached. Now for all these things, this is very useful. So the natural protein adhesives are prepared with the help of this biotechnology.